went through all of your the comments that you gave back on the mid-semester evaluation. So thank you for those between the group who did them last week and then you guys in class this week. So there were a few like common themes that I picked out through them. So I'm going to try to make some of those changes. Some things I can't change, like the amount of material. It's I try to pare it down to what I think is necessary, but I can't really pare it down much more at this point. So I keep that in mind as I make each lecture. It's just a lot of material. AMP is a lot of material. We know a lot about the human body, which is good, but it means that you guys have more to learn. Okay, so let's see. Let me share this with you guys. You guys, a lot of people said they'd like more review time, like before an exam or just as we're going through a unit, just kind of stop and review, which makes perfect sense with all of the material we have. Time-wise, I have to limit that a little bit just so we get the material in. But I'm going to try to squeeze in or fit in some more review time in the class. So on that note, I want to talk about some changes that I'm going to make to when your next test is, basically. So it's scheduled right now for next Wednesday. Yeah, so a week from today. Today's Wednesday, right? Yeah, a week from today. I'm going to move that actually a whole week later. So I'm already going to be, it's going to be tough to fit in the muscular system by the end of Monday before the exam. And I also got comments that you guys wanted maybe a little bit more time in between the end of a unit and the test. So I'm going to see how I can make that happen, but this will be kind of an easy test at that. So I'll update this calendar, but exam 3 is no longer going to be on the 31st of March, which is where it's scheduled right now. I'm going to move it to the 7th of April. I don't want to do it after the long Easter weekend. I think that's a little bit cruel. So we might get into nervous tissue a little bit. So the nervous tissue stuff won't be on this exam, but in order to sort of not waste too much time, maybe get into the nervous tissue depending on where we are review wise and on the muscular system before the test on Wednesday, April 7th. So I'll update that on here and then I'll update it on the assignments page so it shows up on your like upcoming or to do or whatever it is on Canvas. If that's a major issue for anyone, please let me know. What else? So yeah, I'll put in the days I'll have set for review. So Monday we'll basically be doing another Jeopardy for the muscular system. So a lot of people said they like the Jeopardies for the bonus points on the exam, as well as to help review rather than me lecturing, very boringly about all the muscles or the bones in the body. So we'll do that again on Monday, and then Wednesday, if I have to kind of wrap anything up with the muscular system, I'll do that, but we'll also kind of start reviewing some of the material. So whether it's the muscular system or muscular tissue or the joint stuff, I'll kind of figure that out as we go, but we'll do some review on the 31st of March, and then a little bit more that Monday before the next exam, so the 5th of April. I'll do a little bit more review, trying to fit in some more review opportunities. So Jeopardy will keep going. Kahoot quizzes, we'll use those for review activities as well. And you guys also, well, this is for both the classes, so I don't know if this was you guys or the 8 a.m. class, but I saw multiple comments on just having more, and this kind of goes along with review, more class activities, worksheets, that kind of stuff. They're not graded, but at least it breaks up the lecturing. That being said, today I'm pretty much just talking the whole day. So there's going to be some days that are just lecturing. It's kind of tough to break up every single lecture. But on Friday, we'll be doing some of that, kind of having you guys apply what you're learning instead of me just talking about it and then testing you on it. So I'll try to incorporate that a little bit more. I think that's a great idea. And I tried that at the beginning and then kind of got away from it. So it was a good reminder. Let's do that more. The outlines. So I got mixed reviews on the outlines. Some people really like them. Some people really don't like them. Some people find them helpful. Depends on how much effort you put into them, I think. I'm going to keep them as is for now. They're not worth a lot. They are worth some. So that being said, if you haven't turned in any, turn them in. It'll be a good review for the test, for the final. I'll still give you credit for it, even if it's really, really late. I just want you guys to look at the material again on your own instead of just listening to me in lecture. So I don't want your grades to be hurt by not doing the outlines. It's supposed to help at least get you to see the words before I talk about them, because a lot of the terminology is kind of tough. Okay. So outlines are kind of staying as is. I actually saw more positive reviews of the outlines. I have positive circled on my note card here, so I guess that's a plus. I'm glad you guys are, as much as you probably don't want to do them, at least they're hopefully helpful. Yeah, and then I kind of covered that stuff too. So those are the main things I'll try to change. I can't change the amount of material or yeah, basically the amount of material was a lot of the comments. So just in order to kind of comprehend the material a little bit better, I think we'll try to incorporate some more class activities and reviews. So hopefully it won't be, it'll still be a lot, but maybe not as overwhelming. Okay, so I will update this calendar right after class. Oh, and I put the worksheet that we did on Monday, that muscle tissue worksheet, I put the key up in case you guys didn't get all the answers. And I know you guys on Zoom had a tough time kind of seeing what I was pointing at. So I put the key on Canvas. It's available for you. You definitely want to know that material. I'll be asking about that on the exams. So this exam as well as the final. Also, I should say this because I see people on their cell phones, in an effort to keep you guys focused. So a lot of people said they have a tough time focusing. I can't compete with what's on your cell phone, right? What's on your cell phone is way more exciting, or on your watch is way more exciting than what is on what I'm talking about. So I'm gonna try to stay on top of you guys. If you're on your cell phones, just kind of remind you, okay, let's refocus up here, rather than you guys wasting your time in here. Essentially, if you're here, this is basically a 15-minute study period. And I think I said that at the beginning of the semester, and you know, a lot's happened since then. But the more you focus in here, the less you have to study outside of class. So I'm going to try to stay on top of you guys. I'm not trying to be mean. It's more just, okay, focus, and I can only do so much to help you guys, you know, focus in class, and that's one aspect. Guys on Zoom, sorry, it's kind of, I mean, it's a self-motivational thing. I know it's not the best situation. If you do want to be in class all the time, there's room in here. So if that's of interest to you and you think you work better in lecture, you're more than welcome to come or let me know, but there should be plenty of room for everyone if you want to come every day, basically. I don't think that'll be an issue. Okay. I think that is it. Any questions about any of that? Okay. A reminder that your video project is due Friday by 11:59 p.m. If you're having issues with anything or have questions, let me know. I saw, let's see, a question from one group on my email as I was just getting ready for this class about, I don't remember the whole question, but they mentioned like a 3-minute video clip. Is that okay? 3 minutes is a bit long considering it's an 8 to 10 minute presentation. So I would say 2 minutes max. You can have video clips. It definitely makes it a little bit more interesting, but I know 2 minutes sounds arbitrary, but 3 out of 8 minutes, that's almost half of your video is someone else talking about your information. So 3 minutes is a bit long. Absolute max, I'd say 2 minutes. So if you have a video clip, you can kind of pick out the most important part and then just show that piece of it. But video clips are fine. 2 minutes is even a bit long for an 8 to 10 minute, but I'll give you guys that. Yeah, so if you have any other questions, let me know as you are kind of preparing that. Any questions right now? Okay. Back to muscle tissue. I think, did we look at this slide? Do you guys remember? Is this where I ended? I ended a little bit differently in both classes. Well, let's go back one. I think I did talk about this. We'll go back one just so we definitely know what we're talking about here. So I was talking about kind of the setup of what a muscle is made of. I'm pretty sure I went over this terminology, but just as a reminder, muscles basically work on electrical signals, just like nerves. So electrical signals are being sent throughout your body in order to make your muscles contract or your nerves fire. And this change is called, this difference basically across the cell membrane, which is what we're talking about, is called a voltage or charge differential. So voltage is basically kind of, you can think of it as like potential energy. So I've called it electrical potential here. Potential energy is just waiting to be released. Inside the cell, it's a negative charge compared to outside the cell. So that's really important, having a negative charge inside the cell versus a positive outside the cell. And this is maintained by the sodium-potassium pump. I'm pretty sure I went over this. We talked about the sodium-potassium pump a long time ago, like way at the beginning of the semester. But I'll kind of remind you of what the scenario that it sets up to allow for this electrical signal to be sent along a muscle cell. Okay, so here we have a resting muscle cell. So the muscle is not contracting, it's just resting. So unstimulated. The sodium-potassium pump is moving sodium ions and potassium ions across the cell membrane in a very specific way, so that you have more sodium outside the cell, more potassium inside the cell. And this actually is why like Gatorade has electrolytes. That's sodium and potassium. A lot of the electrolytes are just sodium and potassium because you have to have these sodium and potassium ions here in order to create the electrical charge for when you need to contract a muscle. So that's why electrolytes are, they're just salts that are why electrolytes are so important if you're exercising a lot and sweating a bunch. Okay, so sodium outside the cell, a lot of sodium outside the cell. Here we have the cell membrane, a lot of potassium inside the cell. So sodium outside, potassium inside, and then in addition inside the cell, you have a lot more anions, and these are just noted by the A and the negative. So we don't have to worry, let's not worry about what they are specifically, but that creates a negative internal charge in the cell. So negative inside, positive outside. Negative inside the cell, positive outside the cell. So these sodium and potassium ions now naturally basically want to move to the opposite side of where they are, just by basic diffusion. If this gate opens up. So there are gates that will allow sodium and potassium to move down their concentration gradients. And that is what's going to create what we call the action potential. So on the next slide, it's quite a few bullet points, basically lining up what's happening to create this electrical charge on the cell membrane. So I'm going to talk about that, but just keep this image in your head where in a resting cell, lots of sodium on the outside, lots of potassium on the inside. Okay, so when a muscle contracts, when it gets stimulated, neurons are going to release that neurotransmitter, which I think I mentioned on Monday, acetylcholine. And I'll talk about that again today. But without the release of acetylcholine, or ACH, from a nerve cell, you don't get muscle contraction. So it's a really important neurotransmitter. It's just a chemical that sends a signal to the muscle. So that's the very first part of creating this electrical wave that's going to be sent down the muscle cell. That acetylcholine will essentially open up gates for the sodium and the potassium in the cell membrane. So first the sodium gates open, Because there's more sodium outside the cell, it's naturally going to want to flow down its gradient into the cell. So sodium is going to go from outside the cell to inside the cell. Potassium will do the opposite because its concentration is flipped. When sodium flows into the cell, sodium is positively charged, right? So it's bringing more positive charges into the cell. That's going to make the internal charge of the cell increase. So go from a negative to a positive. That process, and I should have bolded this, is called depolarization. So depolarization is where the inside charge of the cell becomes positive because of sodium ions flowing into it. Before that, the cell membrane was polarized. There was a difference in charge on the outside versus the inside. So depolarization means now it's just positive across on both sides, basically. So then, like I said, the potassium gates will open after the sodium gates are closed. Potassium rushes out of the cell. That basically turns the cell negative again inside. So they're sort of working at odds with each other, but that's what's creating what we call the action potential, which I'll get to in just a second. So sodium comes in, the inside of the cell gets positive, depolarization. Potassium goes out, the inside of the cell gets more negative because you're losing positive ions. So it comes back down. That's repolarization. So now again, you have negative inside the cell, positive outside the cell. So that really quick kind of up and down, and you can look at this on a, using a machine, kind of like if you imagine an EKG machine or something, you can look at the charge essentially in the cell going up and then going back down, kind of almost not instantaneously, but very quickly. And that's called the action potential. So really quick up and down voltage shift, going from positive to negative, is called the action potential. That's essentially the electrical impulse. That'll be passed kind of along the plasma membrane or the sarcolemma of the muscle cell. So it'll move in a wave down the muscle cell, which is really long. Remember, they can be up to 30 centimeters long. So it'll move in a wave along the sarcolemma. That's not officially what contributes to contraction, but that's the very first stage of what contributes to contraction. You have to have this electrical impulse moving down the plasma membrane. So I'll talk about what happens after that, but understanding how this happens, without this, we don't have any kind of myosin and actin binding to allow for the actual contraction. So here's these two terms defined on their own slide. They're really important to keep separate. The resting membrane potential is just when a muscle is resting. That's where you have a negative charge inside the cell, positive outside the cell. That's that potential energy that can be released when signaled. The action potential is that quick up and down voltage shift. So that's sort of the released energy. You can think of it that way. It travels along the cell membrane as a wave or an impulse. And that wave or impulse is going to lead to, which I'll get into in a minute, the release of calcium ions. So that's the next step, which I won't, it's not on the very next slide, but we'll get into it very quickly here. So that's why we need the electrical signal being sent down the sarcolemma, because we have to have that calcium released. Okay, so resting and action potential. These next like 10 slides, so 10 slides after this, are going to go into excruciating detail about exactly what happens from the start of when that neurotransmitter is released, the acetylcholine. So we're going to start with that point and follow the process through these 4 main phases of muscle excitation, contraction, and relaxation. These are the 4 overall phases. I think across the next 10 slides, there's about 18 individual steps. So lots of detail. And this is what we will look at again on Friday as sort of a review. The more you look at this, hopefully, the more it makes sense and kind of gets ingrained into your head. So first we have the excitation phase. That's where you have the action potentials from the nerve basically being sent to the muscle. So the muscle is getting excited from what the nerve is sending it, which is primarily that acetylcholine. And then the action potential, so that wave along the plasma membrane, is going to lead to myofilaments that will eventually contract. So that's the excitation contraction coupling. So the excitation is the electrical signal. It's getting excited by electricity. That will lead us into the contraction. So this is kind of an intermediate stage you can think of between excitation and contraction. So does anyone remember what the myofilaments are? When I say it's going to activate the myofilaments, what am I talking about? Really important proteins, filaments. There's a thick one and a thin one that you labeled on your worksheet on Monday. Myosin and what's the other one? Actin. Yeah, myosin and actin, those are the myofilaments. So those are what are actually going to bind and then lead to contraction. Myosin and actin you want to be really, really familiar with. Those are the myofilaments. So in contraction, those 2 are going to bind, and then the sarcomere, if you remember that, is going to shorten, or basically the overall muscle cell will shorten, the muscle fiber. So it'll contract because of the actin and myosin binding to each other. And then relaxation is pretty straightforward. Everything just kind of goes back to normal. The muscle fiber extends again back to its normal length. So like I said, this is the overview. You want to know these 4 general phases and kind of what happens in each one. Now we're going to go into detail within each of these 4 phases about what, from a detailed physiology perspective, what's actually happening at the really like within a cell, subcellular level. And I would say this is one of the toughest parts of this whole chapter, just because there's a lot of detail, and it's a lot of it's kind of biochemistry, nothing like complicated biochemistry, but understanding some binding between different elements. So focus as much as you can over these next 10 slides. I have diagrams basically on the next 10 slides from the text. Let me go to the next slide here. So it's going to be diagrams kind of showing you what's happening at a extremely zoomed in level. Down here, these little yellow numbers with the description, those are from the text, like directly from the text with the diagram. I have, in some cases, or in every case, I've kind of described it in my own words. Sometimes it's pretty much the same. So arrival of nerve signal sent to muscle. So I gave a little bit more detail there. If there's more detail in the text version, than what I have. You just need to know what I wrote here. So these text boxes at the bottom are really what you want to focus on as describing these different steps. It helps me to reword stuff in my own words, so I remember it a little bit better. And I think I simplified some things compared to how they explain it in the text. Sometimes they explain it at a kind of, I don't know, too complicated or kind of too high of a level. So I've kind of maybe simplified it a little bit for you. Hopefully that's helpful. All right, so we start with number 1 of 18. Number 1, what we have here is the nerve. So all of this is the nerve. This is where the nerve and the muscle come together. So right here, that interaction between the nerve and the muscle. So for you guys on Zoom, this is all the nerve, this weird shape thing. The nerve is sending an electrical signal as well down itself to the muscle. So that nerve signal is arriving at this juncture between the nerve and the muscle. That's going to lead to the release of these little green dots, which are the ACH. Oh, sorry, those are the synaptic vesicles. These are the ACH, that neurotransmitter, acetylcholine. So acetylcholine is going to be released from the nerve cell when the nerve cell sends a signal down to it to release the ACH. So the ACH is released. It will bind to receptors on the muscle. So these little purple things are membrane proteins. The ACH here are the green dots. So the green dots right here are the ACH. For you guys on Zoom, this is the membrane protein that the ACH will attach to. That's how the signal gets from the nerve to the muscle, by the ACH binding to the receptors. Okay, so that's the very beginning, the excitation of the muscle fiber. Here we're zooming in to look at what's happening at that protein, the ACH receptor. So you have the binding, we already talked about that. ACH is binding to the receptor. And then that is essentially going to allow that sodium and potassium that I mentioned to move into and out of the cell, like we just talked about, that action potential. Sodium's moving in, potassium is moving out. So just the binding of those ACH particles will open those sodium and potassium gates within the protein. It also opens some other gates, which just allow more sodium and potassium to come in or go out. So don't worry about the different gates. They're just kind of different proteins that allow sodium and potassium to move across the membrane. The take home here is really that sodium's moving into the cell, potassium's moving out after that ACH binds. And then that creates the action potential. So that's the action potential we talked about, where you have that depolarization, repolarization, up and down shift of voltage across the membrane. So think of that as basically electricity being released. Action potential, think of it as like an electrical impulse. So that electrical impulse, so now we're zooming out a little bit. Here's the nerve where it's attached to the muscle. So here you have the membrane of the muscle cell. Here, right along here. These orangish yellow arrows are the action potential. So it's not well labeled, but the orangish yellow arrows that are pointing to the left here are the action potential. That's that impulse, that wave of electricity being sent along the plasma membrane. So just imagine a wave of electricity coming from the nerve muscle juncture. That will flow down into the T tubules, which I mentioned briefly when I was going over the anatomy of a muscle fiber. The T tubules are just infolding, so just kind of a channel in to the cell. The electrical potential, the action potential will flow down into there. So this electrical signal flows down into the T tubule. That is going to release calcium ions from the sarcoplasmic reticulum. So I know it's a lot of detail about a lot of really tiny anatomy, which is why this is sort of one of the toughest parts of this chapter. So for you guys on Zoom, if you couldn't see, the electrical signal is being sent down the T tubule. These little white dots are the calcium ions. That electrical signal leads to the release of calcium ions out of the sarcoplasmic reticulum. And here they call it a terminal cistern. It's just like, that's a very specific part of the sarcoplasmic reticulum. You don't have to know that term. That's why I put sarcoplasmic reticulum here, so you're not confused. All right, calcium is released. That's huge. That's what we need. We need calcium to allow for muscle contraction. And you can see the filaments here. Maybe you might not be able to see from back there. So here we have the thick or the myosin filament, the thin actin filament. So we have sarcomeres with those Z lines on either side. So here's the thick filament and the thin filament. It's hard to see on the screen. So that's where the calcium is flowing to. It's flowing to these filaments. And what it's doing there, so we're going to zoom in a little bit further here into specifically the actin. Well, we have some myosin over there. So zooming into the myofilaments, those actin and myosin protein filaments that we just talked about. So what the heck does calcium do here? It is going to bind to troponin, which is this weirdly shaped cone-like thing on actin. So troponin is on actin. The calcium has to bind to troponin. So calcium to troponin, this should say +2, sorry, Ca +2, not that it matters all that much. When the calcium binds to the troponin, it's going to shift this other molecule, tropomyosin. Calcium binding to troponin is going to shift the tropomyosin off of the active sites, which now you can see over here on the right of the actin. So just the calcium binding to the troponin will shift the tropomyosin off of the active sites. Those active sites need to be exposed so the myosin can bind. If the active sites aren't exposed, the myosin can't bind to the actin and you don't get contraction. This seems like kind of a complicated, roundabout way to get to a muscle contraction, but this is where evolution has left us. This is how it works. Many steps to allow for this muscle contraction. Okay, so you want to know that calcium binds to troponin, it moves tropomyosin, that exposes the active sites, so myosin can bind. Now we're finally going to get into the contraction part. So this was all leading up to contraction. We haven't done any contraction yet. Okay. So we need some ATP to get the myosin head in place to bind. It's not really an individual step on its own, but just know that you need ATP to make the binding happen, because we got to move the myosin head into place. That's going to create, when they bind together, so the myosin binds to the actin, the active site of the actin, when the myosin binds to the actin, you create a cross bridge. And I've bolded that term because it's an important term. A cross bridge or a bond between the actin and the myosin filaments. We still haven't contracted it all yet. We're just binding. Nothing's moved. The next step, we're finally going to contract and move one of these filaments. Does anyone remember which of these filaments, the actin or the myosin, actually moves during contraction? Like which one's being pulled? Actin, but thank you for answering. Yeah, so the actin is the filament that's moving. That's a really important concept. So think about it as the myosin is grabbing on and then pulling the actin filament along, whatever way it's going. We'll imagine, I think on the next slide it goes that way. So the myosin head binds to the actin and then shifts the whole actin filament towards the center of the sarcomere. So here you have the myosin head, it's going to bind and then shift the actin filament. So the myosin filament, the head moves a little bit. So that's fair, there is some movement, but the whole filament doesn't move. It just kind of pulls the actin filament along. Okay, let's do that. So now the actual physical contraction. Yeah, for some reason, the text puts the first step here and then second up here this time. It's confusing. But myosin, like I said, is going to pull the actin filament along. It will then unbind and rebind further up. So it'll unbind from the actin, bind to another active site, and shift the actin a little bit further along. So here you have the myosin head binding to the actin. Once it's shifted it as far as it can, it will unbind and then rebind further down the actin filament and just kind of keep stepping along the actin filament like that. There have been a number of models over the years kind of trying to explain this whole process. And this is the one that is mostly accepted nowadays in the scientific realm. It's called the sliding filament model of muscle contraction. So you want to know that term for this whole, like when we started at the very beginning with the excitation phase, this whole process, this whole model is called the sliding filament model because the actin filament is sliding along. Okay. So that's step 13 of 18. We're almost there. So now the relaxation phase. Everything kind of needs to go back to where it was before we started this whole process. First, the nerve needs to stop sending a signal to the muscle to contract, right? That's kind of the first step in relaxation. Stop releasing ACH. If ACH is not being released, you don't have the binding of ACH, the protein, the receptor protein, which means you don't have the electrical signal created, which means you don't have the calcium being released. So that has to happen first. No more ACH being released. It gets degraded. You don't have to worry about the name acetylcholinesterase. That's just the enzyme that'll break it down, but don't worry about that. Just know it is broken down, it's released and broken down. The calcium that was released from the sarcoplasmic reticulum goes back to the sarcoplasmic reticulum. So it is essentially just stored here until it's needed again. So it'll unbind from the troponin, which I'll show you on the next slide. It'll unbind from that and move back to the sarcoplasmic reticulum until it's needed again, until another nerve signal is sent to start this whole process over again. All right. So yeah, calcium unbinding from troponin. It's going back to the sarcoplasmic reticulum like I just mentioned. When the calcium unbinds from the troponin, that makes the tropomyosin shift back to covering those active sites. So now myosin can't bind to it anymore. So calcium being released, that's going to make tropomyosin move back to cover the active sites. So now contraction isn't possible because you can't create this cross bridge between myosin and actin. And that's it. That's 18. So that's the very end of that whole process of a muscle contracting. Like I said, in excruciating detail, probably more than you ever wanted to know. But when you think about it, every time you move any muscle, this happens. So you move your eyeball, and this whole process, all 18 steps are happening almost instantaneously. Every time you want to move your finger or walk, this is all happening to a bunch of different muscles at one time. So it's pretty amazing how quickly it can happen. Okay, so move into something that's a little bit more big picture. So we're moving away from the super subcellular view of muscles. So for the rest of this lecture, I'll have to finish it on Friday, but the rest of this lecture is, I'll talk about ATP a little bit, so a little bit more biochemistry after this, but for the most part, kind of bigger picture, I guess, bigger picture stuff with muscle tissue and kind of how the muscle tissue works. So have you guys heard of isometric workouts, like isometric lifting? Yeah, so that's kind of a big deal these days. The opposite of that is isotonic. So your muscles can contract in an isometric way or an isotonic way. Isometric just means, so both of these, the muscle's contracting. There's tension on the muscle. Isometric is where the muscle doesn't change in length. So it's almost like you're pushing on a wall that's not moving. Your muscles are contracting, but the length of the muscle isn't changing because you can't actually move that wall. So that's isometric. Isotonic is the opposite. You have muscle contraction and tension, but the muscle does change in length. So if you're lifting a weight with your arm, like this lady. So if she lifts the weight up and then moves it down, her bicep is changing in length. When she flexes her arm and brings the muscle up, that's called concentric contraction, and the muscle is shortening. So your bicep actually shortens, or any muscle shortens when you flex that muscle for those body parts. That's concentric. Eccentric is the opposite way. So your muscle is lengthening. You're lowering the weight, for example. So here, she's lowering the weight. That's eccentric contraction. So the muscle lengthens, but there's still tension on it. So shortening versus lengthening. Those are the two isotonic muscle contractions. Here, her isometric is just no movement. She's just holding the weight there. So there's tension on the muscle, but it's not changing in length. All right, and I'll go to this next slide. And just kind of introduce this. I don't have time to go through this concept today. But next we're going to talk about how our muscles use ATP. Basically talk about when you start exercising up to and going through the process of aerobic respiration in your muscles to anaerobic, back to aerobic. So your muscles go through these kind of three main phases as you exercise, kind of going into, it probably gets you to 40 to 60 seconds into exercise. Your muscles go through both aerobic and anaerobic respiration. So we'll talk about this and how that produces ATP. So if you remember, ATP is the main energy source for cells, right? So ATP is that fuel for cells. Muscles need a lot of fuel because they do a lot of activity. So these two main pathways of ATP synthesis are what muscle cells will use depending on how much oxygen is available. If there's not enough oxygen, they'll do anaerobic respiration or fermentation. And that's going to produce lactate, which will create lactic acid basically. That's what makes your muscles sore the next day if you worked out really hard, because it's an acid. That has to be gotten rid of by the liver, so detoxified by the liver. And it doesn't require, or it doesn't produce nearly as much ATP. So this has to happen sometimes when muscles don't have enough oxygen, sorry, anaerobic does. Ideally, your muscles are doing aerobic respiration for the most part. You get a lot more ATP, a lot more fuel out of it. And all of this is due to basically breaking down organic energy sources. So organic energy sources, glucose is usually what we think of. So glucose, you can break down obviously fats, fatty acids, proteins. All of those will go into the process of respiration and produce ATP, just in different ways. So yeah, we'll talk about those different phases on Friday and how that happens over the course of a workout. Okay, and then we'll finish this up on Friday, get into the kind of an intro to the muscular system as well.